Hello everyone, my name is Nrf2. I'm from the Graduate School of Medicine, Kobe University, Japan. The title of my presentation is Activation of Nrf2 ERE signaling pathway upon hepatitis B virus infection exert an inhibitory effect on viral replication. So HBV and redox homeostasis. HBV infection-induced disorder of redox homeostasis is associated with the development of hepatic diseases. HBV enhances reactive oxygen species production and also induces expression of antioxidant enzymes. However, the mechanism of HBV-induced antioxidant response remains unclear. So, this study aims to clarify the molecular mechanism of HBV-induced antioxidant response and to determine the role of antioxidant response in HBV replication. Here, HBV infection significantly increased ARE luciferase activity. So, from this result, we can say that HBV infection induces antioxidant response. Next, which viral protein is required for HBV-induced enhancement of ARE luciferase activity? Here, we can see that HBx specifically enhances ARE luciferase activity. And then here, HBx derived from HBV genotype A, B, C, and D enhance ARE luciferase activity. Importantly here, HBV wild type can induce ARE luciferase activity, but HBV mutant delta HBx does not enhance ARE luciferase activity. From this result, we can say that HBx protein plays an important role in HBV-induced antioxidant response. To clarify molecular mechanisms of HBx-induced ARE luciferase activity, we focus on transcription factor Nrf2. Nrf2 is a crucial regulator of antioxidant defense. Here, in normal condition, Nrf2 will bind to the KEAP1, leading to ubiquitination and degradation of Nrf2. In a stress condition, ROS will be produced and disrupt the interaction between Nrf2 and KEAP1, leading to stabilization of Nrf2 and move to the nucleus bind to the ERE promoter to produce the antioxidant enzymes. To determine whether Nrf2 is involved in HBx-induced ARE luciferase activity, we knock down the Nrf2 mRNA expression by using the siRNA. Here, we can see that knockdown of Nrf2 strongly decreased HBx-induced ARE luciferase activity and restored by siRNA-resistant Nrf2. This result suggests that Nrf2 is involved in HBx-induced ARE luciferase activity. Next, we ask a question. Does HBx activate Nrf2, leading to enhancement of ARE luciferase activity? To clarify the role of HBx in the activation of Nrf2, we examine the Nrf2 protein expression and Nrf2 nuclear translocation. Here, we can see HBx enhances endogenous Nrf2 expression. In the nuclear extract here, we can see that HBx promotes nuclear translocation of Nrf2. In Nrf2 expression alone here, we can see that Nrf2 is located in the cytoplasm. But when Nrf2 co-transfected with the HBx, Nrf2 will move to the nucleus. So from this result, we can say that HBx enhances Nrf2 expression and nuclear translocation. Then, how does HBx induce Nrf2 activation? In normal condition, Nrf2 will bind to the KEAP1 in the DGR domain, and then leading to degradation of Nrf2. So here, we hypothesize that HBx interact with the KEAP1 in the DGR domain too, and blocks the KEAP1 and Nrf2 interaction, thereby inhibiting ubiquitin-dependent degradation of Nrf2, leading to the stabilization of Nrf2, move to the nucleus, bind to the ARE promoter to produce the antioxidant enzymes. Here, we did immunoprecipitation assays. HBx, but not HBc, interact with the KEAP1. And here, we can see that DGR domain of the KEAP1 is responsible for the interaction between HBx and KEAP1. So from this result, we can say that HBx and Nrf2 competitively interact with DGR domain of the KEAP1. Finally, does Nrf2 affect replication? To To clarify the role of Nrf2 in HBV replication, we examine the effect of overexpression of Nrf2 on the HBV core promoter activity, HBV total RNA, and pgRNA expression levels. Here we can see in the figure A, Nrf2 strongly inhibits HBV core promoter activity. And in the figure B, we can see here that Nrf2 significantly inhibits HBV RNA and pgRNA levels of the HBV genotype A, B, C, and D. So from this result, we can say that Nrf2 inhibits HBV core promoter activity, leading to downregulation of the pgRNA levels. Downregulation of the pgRNA levels by the Nrf2 would further decrease the HBC protein expression and inhibits the amount of the extracellular HBeAg. So, in the summary, a proposed model for activation of Nrf2 ARE signaling pathway against HBV infection in the normal condition, Nrf2 bind to the KIP1, leading to ubiquitination and degradation of Nrf2. But in the HBV infection, HBx will interact with the KIP1, then leading to stabilization of Nrf2, move to the nucleus to bind to the ARE promoter to produce the antioxidant enzymes. Importantly, Nrf2 may be bind to the ARE-like motif in the cccDNA and strongly inhibits the core promoter of the HBV and downregulate the pgRNA. So our study suggests that activation of Nrf2 ARE signaling pathway is a therapeutic strategy against HBV. I think that's all. Thank you very much.